Hello and welcome to all my Nexus students. So, how are you all? I just hope that you all are doing fine and must be studying well. So, finally, today we are going to start with the class 11th syllabus, and especially for botany, I am going to start with the new chapter, that is cell, the unit of life. This is the first chapter that we are going to cover up, followed by cell cycle and cell division. Right? So what are we waiting for? Let's directly start with the Nexus batch. One thing let me tell you, students, see, since this is a recorded form of batch which will be given free to you on the PW app as well as on the YouTube channel of PW English. So if you have any doubts, feel free to post in the comment section, and yes, you will get the responses. Second, beta, try to make the notes along with the class. Already you will get the notes in the form of PDF. Whatever I am going to write, that will be given to you, but make sure that you are making your own notes as well, so that even if later you want to study, you can do that. Right? Chalo. So let's start with the lecture number 1 of the chapter Cell, the Unit of Life. Today's lecture is going to be a basic one, in which we are going to study what is cell, who discovered cell, types of cell, etc. So today's lecture is going to be a simple and short one. Hope you all are going to enjoy. Let's start. Okay, so starting with the introduction portion. In introduction, we are just going to discuss the name of the chapter itself. You know, chapter name is all about cell. So first question that arises in mind is, what is cell, right? So who is going to define this for me? I hope everyone already knows the answer. What is cell? Yes, cell is the basic structural and functional unit of life. All living organisms are made up of cell or product of cells like tissue, organ system, etc. Right? So that is why we say that cell is the basic unit of life. Any organism who is less than a cell is not considered living. To be considered as a living object, body should be at least made up of one cell. A single cell is also capable of performing all those activities which a multi-celled organism can do. Right? I hope all these points are clear. If yes, then please make up the notes as well. So cell is the basic structural and functional unit of life. So cell is the basic structural and functional unit of life. All living organisms without any exception are made of cells or their product are made up of cells or their product. Then even a single celled organism is capable, single celled organism is capable of performing all metabolic activity which a multi-celled organism can do, which a multi-celled organism can perform. Right? Understood, everyone? So this is the first point, just underline it. It's the basic structural and functional unit of life. Second important point is, All organisms are made up of cell or product. Third point. So even a single celled organism is capable of performing all the important metabolic activities, right? Now question arises, who discovered cell? Hence, I hope you all know the answer, that the first cell was discovered by Robert Hooke. He actually coined the term cell while studying on a thin slice of a cork. You know cork, if you have seen a champagne bottles and all, then those champagne bottles, they are fitted with the cork. So he took that cork, cut a thin slice, viewed it under a simple microscope, and when he viewed that thin slice of a cork under the microscope, he saw some honeycomb-like compartments which are called as cella by him. Cella means cell. So from here, the word cell came up, right? So what is the next point you are going to write, beta? Term cell was coined by Robert Hooke, right? He was working on which organism or which structure was he working? He was working on a thin slice of a cork. Right? Now, while he was working on a thin slice of a cork, he saw some honeycomb-like compartments. So just add a page to continue writing. Now, he observed some honeycomb-like compartments like this, beta, right? Which he termed as cella. Cella means cell. So these are basically honeycomb-like compartments. This was the view of a thin slice of a cork while this fellow Robert Hooke was viewing the thin slice of a cork, right? Now, you know, cork, it's the dead part of the plant body. It's a dead material. So basically Robert Hooke observed the dead cell, right? No doubt he coined the term cell, but he observed the dead cell. To be more specific, he observed the cell wall, right? Now, then question arises, who saw the first living cell? If Robert Hooke saw the first dead cell, or to be more specific, cell wall, then who observed the first living cell? Then here is your answer, Antony von Leeuwenhoek. He was the scientist who saw the first living cell while working on mud water. He was working on mud water. He took the mud water, kept it under the microscope, and he saw some moving structures which he called it as animalcules. And these animalcules were rich in bacteria, protozoans, and all those single-celled organisms which are very richly found in soil, right? So from here came the first living cell that was discovered by Antony von Leeuwenhoek, right? Got my point? Shall we write it? Come on. However, the first beta living cell was discovered by AV. AV stands for Antony von Leeuwenhoek while he was working with mud water, right? Clear? Okay, very good. Now, you are a multi-celled organism. You know, your body is made up of so many cells which joins together to form tissues. Tissue give rise to organs. So you know different levels of hierarchy. So if I say that cell is the basic unit of life, right? Now, in a single-celled organism, of course, cell is the only part which they have. But if I talk about a multi-celled organisms like you and me, then our body is made up of zillions and millions and trillions of cells. But they don't remain as cell. Rather, they join, they unite together to form the next level of body organization called as tissues. which later joins to form organ. Organs, they join to form organ system. And finally, organ systems, they reunite to form a multi-celled body called organism, right? So these are the different levels of hierarchy. Correct? Got my point, everyone? Done? Okay. Now, beta, question arises, like in a multi-celled organisms like you and me, our whole body movement, right now I am talking, I'm walking, I'm using all my senses, so definitely someone must be controlling all my movement. And which is that part of the body that controls all the body movement? Brain, right? Similarly, if I consider a single-celled organism like amoeba or bacteria, do they have brain? Or even without brain there, all functions are getting regulated? So answer is yes. Every individual cell has its own brain, which is called as nucleus of the cell. So every cell has its own brain. See, of course, a whole body has an extra organ as called as brain. But if I talk about individual cell, then every individual cell also has to regulate its own activity, and then they join together to form tissue and then organism like this. And once a whole organism is ready, then comes the brain as one of the organ, right? Clear? Similarly, if I talk about single cell, then all single cells have their own brain, which is called as nucleus, that is responsible for controlling all the metabolic activities, and it was discovered by Robert Brown. So the next point that you're going to write is nucleus, which is actually brain of the cell, that regulates, coordinates all the activities taking place inside the cell, was first discovered by Robert Brown, right? So now so far I have introduced three scientists named Hooke, Leeuwenhoek, Brown. Hooke is responsible for discovering the first cell, which termed to be as a dead cell. Leeuwenhoek is credited to discover the first living cell, and Robert Brown is credited to discover the nucleus of the cell. Correct? Chalo. So this is the basic history related to the cell, which you all should know. Moving towards with the next topic, that is cell theory. There were two scientists, Matthias Schleiden and Theodor Schwann, in the year respectively 1838 and 1839. These two people, Matthias Schleiden was a German botanist, whereas Theodor Schwann was a British zoologist, right? These two individual people, beta, working in two different countries in two different time, one working on plant cell, another working on animal cell, they both found common results. Both the people found that be it a plant body or be it an animal body, they both are made up of cells. So every living organism, from there came the statement, from the work of Schleiden and Schwann, we came to know that yes, cell is the basic unit of life. All living organisms are made up of cells or their product. Whatever activity I am doing right now, my food is getting digested, my liver is working, my kidney is working. So whatever metabolic activities are going on in my body is actually the outcome of cellular interaction. Correct? So these two people actually told us about these points related to the cell, which together is known as cell theory. Correct? Got my point? So now what you're going to write the next? Cell theory formulated by Theodor Schwann and Matthias Schleiden. states that, but who was Theodore Schwann? He was a British zoologist who in the year 1839, whereas Matthias Schleiden was German botanist in the year 1838. Formulated. So cell theory was formulated by these two people, which states. So now write all the 3 points which I told you. Number 1, cell is the basic unit of life. Right? Number 1 point says that cell is the basic unit of life. Second, all living organisms are made up of cells or their product. Right? Third point, whatever metabolic activities we are performing is actually the outcome of cellular interaction. So third point says the metabolic activity is the outcome of cellular interaction. Is the outcome of cellular interaction. Correct? So got my point, everyone? Yes or no? Cool enough. So these 3 are the important postulates of cell theory. Now apart from this is the common thought of both the people. Apart from this beta, Theodor Schwann also stated one more additional point. According to him, all plant and animal, they are made up of cells and their product. However, if individually talking about plant cell, then plant cell had an additional layer outside the cell membrane called as cell wall. So according to, we know that both plant and animal cell, they have plasma membrane. However, plasma membrane is the only layer around the animal cell. But when it comes to plant cell, then along with the plasma membrane or cell membrane, they have one more additional layer called as cell wall. So this was clearly stated by Schwann. So if question comes, which scientist stated that plasma membrane is the delimiting layer in animals, whereas in plant cells they have one more additional layer by the name cell wall? So your answer will be Theodor Schwann, right? This is very, very important. Make sure that this is also written in your notes, as it is very clearly stated in NCERT. So now I am writing it. Possible, write along with me, right? Chalo. So what are you going to write the next? You're going to write that Theodor Schwann stated that animal cell possess only cell membrane as the outermost layer, whereas plant cell also have cell wall as an additional layer outside cell membrane. Right? Got my point, everyone? So this is about cell theory, which you have to remember. Hope you all are going to make the note of it. Now, this cell theory was definitely proposed by 2 people, and they gave so many points due to which we actually can make out what is cell and why we consider that all living organisms are made up of cell. So now the idea is clear that this concept came from these 2 people. But somehow this theory was yet to be completed. This theory was yet incomplete. Why? Because in this theory, beta, it is not mentioned that from where the cells are arising, who is giving birth to the cells. So this confusion was cleared by scientist Rudolf Virchow, who modified the cell theory stating omnis cellula e cellulae. That simply means all the cells are arising from the pre-existing cell. None of the cell is coming from itself. De novo, सारे cells खुद से नहीं आ रहे हैं. Someone is giving birth to them. And who is giving? Their parent cell. Right? So next you are going to write over here is that Rudolf Virchow modified the cell theory. Modified the cell theory and stated, what was the statement he gave? You all know, I think, omnis cellula e cellulae. The statement simply means, now what is the meaning of this statement? The statement, beta, simply means that all cells arise from pre-existing cells. All cells arise from pre-existing cell. That means if this is my parent cell, it is going to divide to produce new daughter cells. Then these daughter cells are going to mature, which they are going to further divide. And like this, every cell has its parental lineage. Correct? None of the cell is arising de novo. Done? Understood? So this much you have to remember for what is cell and definition of cell, cell theory. This is the introduction to the chapter. I hope everyone has understood it. Proceeding further in the chapter, now we are going to learn about the overview of the cell, in which we are going to talk about the shape, size and function. So, you all are science student. You know that your body is made up of different types of cell. Cells can be of different shapes, sizes and function. Some cells are long, tube-like. Some cells are spherical, ball-like. Some cells are cuboidal. Some cells are columnar. So there are so many different shapes and sizes also. Some cell can be as small as 0.3 micrometers. Some cells can be as long as 7 micrometers. So it's not actually fixed. It's not a fixed entity that, okay, cells always have to be ball-like or cell has to be of this specific diameter. No. Cells shows great diversity when it comes to their shape, size and function. अलग-अलग shape के होंगे, अलग-अलग size के होंगे, and their functions are also decided accordingly. Correct? So now what next you are going to write? The next heading is overview of a cell, in which I am going to talk about the variety, the diversity of cell in terms of shape, size and function. Correct? So what are you going to write for the overview of a cell? Cells exhibit great diversity in terms of shape, size and function. Shape, cells shape are highly variable. They can be either columnar, round, polygonal, elongated, cylindrical, etc. Whereas sizes, they also show great variation. In generally, if I talk about a general bacteria, then the general size of a bacteria is 0.3 microns. For example, bacteria is one of the smallest organism who has a size maybe of 0.03, or you can say 0.3 to 0.4 microns. Then the cell can be as long as the longest cell is your neuron, that is your nerve cell. Smallest cell, even smaller than bacteria, is PPLO, which is I think I'm from 0.01 microns. That is PPLO, also called as mycoplasma. Then the largest cell isolated so far is the ostrich egg. Right? Clear? So here you see beta, there are so many variations when it comes to the shape and size. Now let's talk about the function also. See, function, there's one simple line which you have to understand, and it's really very easy to understand. Correct? It is said that whatever shape is of the cell, accordingly the function it is going to perform. जो भी सेल का शेप होगा उसी के अकॉर्डिंग उस सेल का फंक्शन डिसाइड होगा. So in short I can say the function cell performs is according to its shape. So what you're going to write over here? Cell's shape decides the function it performs. Decides the function it performs. Right? Clear? Now, ma'am, can you just elaborate this statement? Okay, the statement is clear in itself, but it would be better if you elaborate this statement for us. Let's do it. Let's try to understand this statement with the help of a very easy example of trachea. Chalo. Now, what is trachea? Who is going to tell me what are tracheads? Tracheads, ma'am, they are one of the component of xylem found in plants, and xylem are responsible for conducting water and mineral. Yes, of course, xylem are responsible for conducting water and mineral, and one of their component is trachea. So, in short, we can say that trachea function trachea's function is conduction of water and mineral. Right? Clear? Yes or no? Okay. Now, before I ask you about the shape of trachea, let's come to our homes. In our home also, you have a tanki placed at the top of your house, and below somewhere under the ground you have a motor, right? You switch on the motor, then that motor picks up the groundwater, and with the help of pipes, you know, you have to lay down the pipes, PVC pipes, and then those pipes, they carry the groundwater from, with the help of the motor, planted downward to the tanki, which is kept up at the top of your house. Now, water is underground. Tanki, the tank, is above your house. So, who is connecting and who is taking that water from the groundwater, from the ground to the top? Of course, the pipes. So, the main function of the pipes is conduction of water. Now, can you all guess what is the shape of the pipe? You must have seen either in the form of photograph or in your homes itself. Pipes are always cylindrical. Have you ever seen any conducting pipe triangular, circular, rectangular? No. They are always cylindrical. Why? Because the function of the pipe is to conduct water, and conduction will be most efficient if the shape is cylindrical. See, according to the function, shape is decided. Function tha conduction ka, so the shape became cylindrical. Correct? So what the statement I gave? That shape decides the function, or according to the function, cell shape has to be decided. So same goes over here. These tracheids are the pipes basically. Tracheids are nothing. These are just the pipes which are responsible for conducting water and mineral. So what should be their shape? If this is their function, then what should be the shape? Cylindrical. In short, they should be long, elongated. So yes, according to the function, shape of the tracheid is long and elongated, and like this they are kept one above the other. These are the tracheids which are kept, they are long, elongated, they are kept one above the other so that their function of conducting water and mineral can be done efficiently from the root to the leaf. Right? So what is the function? Accordingly, their shape is decided. This is one of the example. Second example I can give you is of RBC. RBC is red blood cells, erythrocytes. Now, what is their function? You all know, carrying oxygen to different parts, right? Now, their function is to carry oxygen to different parts of the body in the blood, right? Now, for carrying oxygen, they need to bind oxygen, correct? Now, if they have to bind oxygen to their surface, then they should be in such a shape so that at one time they can carry maximum oxygen. And whose surface area is maximum? Of sphere. So these RBCs are biconcave disc-like, almost spherical bodies which provide maximum surface area to attach maximum oxygen at a time. Correct? Got my point? So their function is to carry oxygen, hence their shape is biconcave disc-like with an average diameter of 7 microns. They are like this. Diameter is 7 micrometers, right? Now to this maximum surface area exposed, now at a time they can bind with maximum oxygen, which is 4. So at a time, each RBC carry 4 moles of oxygen attached to their hemoglobin from one part to the other. So now see, according to their function, their shapes are decided. Correct? Got my point, everyone? So this is the simple overview which we have discussed for the cell. Now, depending upon the nature of the nucleus, you know, nucleus is the main brain of the cell. Structurally, if I talk about, then each activity of the cell is under the control of the nucleus, which was discovered by Robert Brown. Clear? Now, depending upon the nature of this brain, depending upon the nature of the nucleus, type of the nucleus, we can broadly divide cell into 2 categories as eukaryotic cell and prokaryotic cell. Correct? Chalo, let's discuss that. So next heading you're going to write is, beta, depending upon the nature of nucleus, right? So yes, depending upon the nature of nucleus, we have 2 types of cells. Prokaryote and eukaryotic cell. Correct? Now let's try to decode. We will do the difference in detail. But before doing that, let's try to decode the word over here itself. Beta, pro means primitive. Pro, primitive, old, purana. Karyo word we use for nucleus. The biological, the scientific term that we use for nucleus is karyon, right? So karyo means nucleus, pro is primitive, purana. U means true. Karyo again means nucleus. So the cells with primitive, old type, not so well developed nucleus are called as prokaryote, whereas cells with most advanced, well developed nucleus are called as eukaryotic cells. So cells with primitive, purane type ka or not so well developed nucleus. Not so well developed nucleus. So such cells are called as prokaryotic cell, right? Whereas eukaryotic cells are those in which have well developed advanced nucleus, right? Got my point, everyone? Very good. Okay. See, whatever, the more differences we are going to talk about related to prokaryote and eukaryote. But one thing I would like to mention before I start with the main differences between the pro and eukaryotic cell, which you might have understood little bit here itself. Be it prokaryotic cell or eukaryotic cell, both the cells, they have a fluid inside them which is called as cytoplasm. And cytoplasm is the main site, or it is the main area of the cell where all this nucleus and all other structures will be present, and all metabolic activities, whatever cell has to perform, is taking place in this cytoplasm, in this fluid. And this fluid is common in both pro and eukaryote. The content of the cytoplasm may vary. Prokaryote may have some different composition, eukaryote may have some different content and composition, according to which their functions will also vary. However, they both will have cytoplasm. Clear? So what is the note point? Both the cells have fluid, beta, called as cytoplasm. And what is the work of this cytoplasm? NCERT writes, it is the main arena for all cellular metabolic activities. Arena, beta, simply means location, site. Correct? Will you all remember this thing? Yes or no? However, the composition of cytoplasm in pro and eukaryote will vary, which now I am going to discuss in the different section. So for that, divide your notebook into two sections like this, as ma'am has already done on a screen. So here I am going to teach you the main difference between prokaryotic cell and eukaryotic cell. I hope so far you all have understood what is cell, definition of cell, discoveries related to cell, cell theory, as well as overview of the cell, cell size, shape and function. So far we have not discussed anything which is very complicated. It's everything very easy. Now little bit complication may come, but if you pay attention, things are really going to go very smooth. What do you call this one? Nowadays the people use the statement very frequently. Yeah, easy peasy lemon squeezy. So if you really want this to happen, please pay attention and try to understand that what ma'am is teaching, and making notes is again very, very important. You know, since this is the recorded form of the batch, all the lectures given to you will be in recorded manner. So making notes is your liability. Please, as soon as you get the PDF, try to make your own notes and revise from there. Clear? Chalo. Coming back to the topic, to the differences, pro and eukaryote. One basic point of difference I have already told you, that pro means primitive, so they possess primitive nucleus, right? And U means advanced, so they possess advanced nucleus, right? Correct, everyone? Chalo, very good. Now, question arises in mind that ma'am, you're just saying primitive, advanced, but what is this primitive? कैसे हमें पता चलेगा कि this is primitive, this is advanced? Of course, you should know this also. What is primitive? How will you identify if I give you a cell and tell you, okay, just take this microscope and view it under microscope and tell me which is pro and which is U? Then you only, if theoretically you know that pro is primitive, U is advanced, then how will you identify? Till the time you don't know it practically also, that what these terms actually means. So now here I am going to tell you their meanings as well. Primitive means, beta, the genetic material. See, nucleus carries genetic material. Genetic material in the form of chromosome. Correct? Now, in case of prokaryotic cell, they possess primitive nucleus. That means their genetic material, which is represented by one chromosome only, is directly present in the cytoplasm and not separated by any membrane. There is no membrane around this single chromosome that separates the cytoplasm and the genetic material. See over here, I'll draw it. Suppose this is my prokaryotic cell, right? So this is a cell. Now this cell will have a fluid enclosed within its membranes. So this fluid is known as cytoplasm, correct? Now inside this cytoplasm itself, there is the presence of chromosome, correct? Now which type of chromosome do you see? Circle shaped chromosome. How many chromosome? Single. How many strand present? 2. Have I drawn it like this? No. How have I drawn it? In the form of strand. 2 strand. So this is called as double stranded, right? So this is double stranded. DS stands for double stranded. How many number? Single. Nature is circle or linear. Linear means straight or circle? Circular. So they have double stranded, single, circular DNA. Also you can call it as chromosome. So this is the nucleus. This is the nucleus. Yay nucleus. This double stranded circular single chromosome is the nucleus of the prokaryotic cell. So isn't it very primitive type? Now, this is diffused in the cytoplasm. That is not separated by any membrane. It is not separated by any membrane. Correct? There is no membrane surrounding the fluid like this. No. It is directly diffused in the cytoplasm. So such genetic material is known as primitive type. And here to primitive nucleus, there is another term used, that is nucleoid or genophore. So such primitive nucleus is also, there's one term given known as beta nucleoid or genophore. Right? Clear? Understood everyone? Whereas when I talk about the true nucleus, suppose this is my cell, this is my eukaryotic cell. Of course, it will also have a same fluid which is called as cytoplasm. Now here the cytoplasm will have a membrane. Now this is known as nuclear membrane, right? And this membrane will now this is the cytoplasm, right? Now cytoplasm got separated now due to the presence of a membrane. A boundary ban gayi. It's simply like there's a home, there's a house inside which there are no walls. Suppose this is my home, right? Now if I don't draw the walls inside, then how many rooms are there? One room, one compartment. However, if I draw the walls, अब मैंने membrane बना दी, मैंने layer बना दी. So now my home is having how many rooms? 1, 2, 3, 4. 4 compartments are there. So this thing what is lacking in prokaryote beta. In prokaryote, cytoplasm is not divided into any compartment. Why? Because there are no membranes present. So due to the lack of membrane, cytoplasm is continuous. It's just like one open room where everything is present here and there only. However, now if I make the membranes inside the cell, then I can say separately, okay, this is my drawing room, this is my bedroom, this is my kitchen, this is my bathroom. Correct? So here in eukaryotic cell, there is compartmentalization, there is membrane system present, because of which cytoplasm is not continuous. There is separate rooms, one is bedroom, one is drawing room, one is dining room, etc. But here, everything is present in the same room. So you can't separate, okay, this is the bedroom, this is drawing room. No. Here, everything is continuous. Cytoplasm is continuous. But here, cytoplasm is discontinuous. Discontinuous means it is separated or it is invaginated by membrane system. Correct? Got my point, everyone? So here, nuclear membrane is present which encloses double-stranded but linear, straight genetic material called as chromosome. Correct? Here it was circular, here linear, like a line, lambe lambe. Double-stranded, double-stranded. Here one, here more than one. Got my point? So nuclear membrane encloses double-stranded linear DNA or chromosome whose number is more than 1. Got my point? Such genetic material is known as true nucleus. So this is true nucleus. This is nucleoid, this is nucleus. Got the point? Got the difference between advanced and primitive? Yes or no? Beta, primitive is also known as naked. There is another term also used for primitive, that is naked. I hope everyone has understood so far and no one is having any confusion till here. Okay, very good. Now let's proceed further to more differences. Now second difference, you can make it out from the first itself, as here I have told you that in prokaryote, cytoplasm is continuous. It is not separated by any membrane system. So in short, you can say that prokaryote lack membrane system. But In eukaryote, cytoplasm is not continuous. There is the presence of membrane system. So here you can write that eukaryote possesses membrane system. So next point you will write is cytoplasm is continuous as there is no membrane system, or as it lacks membrane system. But for eukaryote you will write membrane system present. Therefore, membrane-bound organelles like nucleus, mitochondria, chloroplast, all are present in eukaryote and such are absent in prokaryote. Hence, membrane-bound organelles like nucleus, endoplasmic reticulum, mitochondria, etc. are found. But what will you write over here? It lacks membrane system, therefore no membrane bound organelle. No such organelles found. No nucleus, no mitochondria, no ER, nothing. The cytoplasm is just like a fluid, continuous, membrane bound organelles, right? Got my point, everyone? Chalo, proceeding further. Okay, now beta, prokaryote may not have membrane bound organelles. प्रोकैरियोट सेल्स के पास चलो माना मेम्ब्रेन बाउंड ऑर्गेनल्स नहीं है, बट कैन दे हैव नॉन मेम्ब्रेन बाउंड ऑर्गेनल्स? यस, ऑफ कोर्स दे कैन हैव. द ओनली ट्रबल विद प्रोकैरियोट इज विद मेम्ब्रेन, एंड इफ देयर इज नो मेम्ब्रेन then such organelles can be found in prokaryote also, like nucleoid, like genophore. Nucleoid is a single genetic material diffused in the cytoplasm. For that, no membrane is required. Hence, it is found in prokaryote. Similarly, non-membraned organelles like ribosomes can be found in prokaryote also, as well as in eukaryote also. So, eukaryote, along with membrane system, they also have some non-membrane bound organelles like ribosome. Prokaryote, they only have non-membrane bound organelles, right? However, non-membrane bound organelles organelle ribosome present, and the nature is 70S. Now, what is this 70S? Right now, I'm not explaining, just writing it over here. As soon as the chapter will proceed, you will automatically come to know that what is this 70, what is this S. So, don't worry. Those students who know, well and good. Those students who do not know what is this 70S, आपको चिंता नहीं करनी है. Ma'am will herself teach as the chapter will proceed. Correct? Now, coming to this beta, here ribosomes are also present, but the nature is not 70S, rather it is 80S. Now, what is 80, 70S? Everything will be decoded, not now. As the chapter will proceed, slowly and steadily, everything will be done. Correct? Clear? Chalo. Now, proceeding with the next point of difference, beta. Prokaryote, now you see, they just have, when it comes to their genetic material, when it comes to their chromosome, they only have one chromosome. And that too, double-stranded circular simples are diffused. So, they need some extra genetic material. Eukaryote, they have so many chromosomes, they have more than one. Like for example, we have total 46 chromosomes. So already we have so many chromosomes, hence in our cells, there is no extra genetic material present. But when it comes to prokaryote, they have the presence of extra chromosomal material called plasmids, right? If you have heard the terminology, well and good. If not, just write it and understand it. So prokaryote cells, beta, they possess extra chromosomal material, that is again double stranded circular DNA called plasmid. In eukaryote, plasmids are absent. They don't have plasmid, right? Correct? Got my point, everyone? Okay, well and good. Okay, so now you see over here that prokaryotes, they have plasmid, whereas in eukaryotes, beta, plasmids, the extra genetic material is absent as they don't need it, right? Because we already have so many chromosomes. What is the need of extra genetic material? Remember it like that. Now question arises, but that ma'am, if prokaryote have these extra, if they have these extra additional genetic material, then what is the role? Because nucleoid is already there, their all metabolic activity is controlled by that single chromosome only. Then what is the purpose of this plasmid? Beta, presence of plasmid in the bacteria or in the prokaryotic cell gives them some extra benefit. Like for example, there is one category of plasmid by the name R plasmid, where R stands for resistance towards antibiotic. So if any bacteria is having R plasmid, whose maybe nature is to show resistance against penicillin. So now if bacteria having that R plasmid is given the exposure to penicillin, that penicillin antibiotic will have no effect on the bacteria. Bacteria will be resistant. So this is the additional benefit given to the cell with the help of this extra genetic material. Correct? So what will you write? Plasmids give extra phenotypic benefits to the cell. For example, phenotypic means morphological, appearance wise, right? For example, there is one category called as R plasmid. There's one type. Like this, there are two more types. When the time will come, I will teach you. For instance, just for understanding, beta, R stands for resistance. Resistance towards antibiotic, right? Resistance towards antibiotic. So any bacteria having our plasmid will be resistant to the attack of a specific antibiotic. So isn't that good for bacteria, right? However, eukaryotes, they lack plasmid, no plasmids for us, as we don't need. We already have so many chromosomes who is taking care of all our needs. But this is necessary in prokaryote, so they have. Right? Clear? Chalo, next difference. When it comes to beta prokaryotic cells, then here I am writing these are comparatively small in size and divides faster. whereas eukaryotic cells are larger as compared to prokaryotic cell, as compared to prokaryotes, and takes time to divide. Takes time to divide. Right? Clear? So this is the next point of difference which you have to write. Now when it comes to examples, except bacteria. Except bacteria, whatever organism you see, protisteans, plant cell, animal cell, all are eukaryote. So next you are going to write as example. So for eukaryotic cell, plants, animals, algae, fungi, protisteans, all are eukaryote. For prokaryote, only bacteria. So if I say that bacteria and prokaryote are synonym to each other, then I will not be wrong. Either you say prokaryotic cell or you say bacteria, one and the same thing, because only bacteria are prokaryotes. All other organisms other than bacteria are made up of eukaryotic cells like plants, animals, algae, fungi, protisteans, etc. Correct? Got my point? So these are the point of differences between the prokaryote and eukaryotic cell. So far today I have taught you what is cell, cell theory, overview of a cell, and types of cells on the basis of their nature of nucleus. Correct? Got my point? So for today I am just going to keep it till here. In our next class we are going to study the detailed structure of a prokaryotic cell and then followed by eukaryotic cell, so on. Right? Okay, cool enough. Now before I wind up my lecture, let's do some questions. So homework will be read NCERT till whatever is taught to you, till whatever being taught today. And please make sure that you complete your notes also. Correct? Chalo. So first question is, who discovered the cell? Very simple. It was discovered by Hooke. As these two people, yes, they gave the cell theory, and Brown discovered the nucleus. Moving with the next, which is the basic unit of life? Which is the basic unit of life? Molecule? No. Atoms? No. Tissue? No. Answer is cell. The basic unit of life is cell that makes up the tissue, and to make cell you need molecules and atom. Correct? So chalo, with this we wrap up our today's session. The first session, I hope you all enjoyed it thoroughly. If yes, then do not forget to mention in the comment section, and please start the studies. Your syllabus has already started. I hope you all enjoyed the session. Thank you for watching me. See you in the next. Till then, bye-bye everyone. Take care.